DNA is the building block of life. The order of genes within it determine every characteristic, from height, intelligence, susceptibility to disease, and whether you like pineapple on pizza. DNA was discovered in 1953, and as genetics developed, CRISPR Cas9 was discovered in 2012. Yet it hasn't stopped people from exploiting it in dangerous ways. CRISPR has near infinite uses and can do more in a few days than evolution could in millions of years. It also poses many ethical and legal challenges to nearly every one of its functions. So how would we allow only those with good intentions to control the future of our species? So what is CRISPR, and what are the benefits and downsides of it? CRISPR Cas9 is a modern technology that allows geneticists to add, remove and alter parts of the genome. It possesses the ability to locate strings of genes, the computer code of life, and reprogram to complete basic tasks, such as editing DNA bases. These are a combination of 4 base pairs: adenine, thymine, cytosine, and guanine. These are paired up A to T and C to G, forming a basic code, like binary but with 4 base elements. CRISPR is commonly introduced via IVF, in vitro fertilization, and CRISPR-Cas9 is made of the enzyme Cas9, which cuts the DNA strand at a specific location by finding the alternate gene code, the one carried on a piece of guide DNA. It is paired with an RNA scaffold, which is used to guide the Cas9 enzyme to the right place to make sure that it cuts only the desired location. Once the cut has been made, the cell recognizes the damage and then tries to repair it with a piece of guide DNA, creating a desired change in the genome. CRISPR is an acronym for Clustered, Regularly Interspaced, Short Palindromic Repeats. It's a type of defense for naturally occurring bacteria which transcribes DNA to RNA upon viral infection, sometimes giving the bacteria memory of that virus and allowing it to protect against future infections. The simple answer is a gene editing technique. CRISPR is a science with many uses, from improving crop yield, engineering humans to be happier and healthier, as well as curing a wide range of harmful diseases, such as sickle cell anemia, and even perhaps cancer. This is done by improving the body's ability to fight back against them through introducing positive genes or removing dangerous ones. Another more abstract use is to slow or prevent the effects of aging by applying genes found in many mammals that slow the decay of the body over time, allowing humans to live longer, healthier lives. In this video, I will explore the dangers and potential issues of it, such as biological terrorism, by creating gene drives to enable mosquitoes to more frequently carry dangerous diseases, ethnic cleansing, and the legal creation of super soldiers for military purposes. Even though CRISPR is a relatively new technology, there have already been incidents of scientists, such as the named Kai, who, accomplishing potential curing of HIV in embryos, did so illegally and without proper safety procedures. This shows how access to CRISPR must be preceded by rules and regulations to prevent dangerous and illegal changes to the human genome. With all these uses and danger, it is clear that CRISPR is a very contentious topic, with both positives and negatives to using it. However, CRISPR will be developed regardless, so it is up to the public and the lead scientists to make sure that CRISPR is a word associated with miraculous breakthroughs in science, and not a name to fear. The aim of this video is to conclude whether it's a tool for good or a danger to humanity, as well as discovering how it can be used to safely improve the quality of life and standard for living for many, as well as explore ways to avoid the dangers that can be caused by the advances in gene editing. Every trait about a person can be found in their genes, from eye colour, height, favourite food, and susceptibility to disease. Each of these traits can be caused by as little as a single gene, to as many as a dozen. These genes can be inherited from someone's parents, or randomly changed by everyday background radiation. Some diseases even, or extreme accidents, can also change them. All these things can change the gene expression while they're being transcribed, which means copied from one cell to its daughter cells. These factors of nature and nurture cause everyone, even identical twins, to have different traits. And it is this which makes the world such a wonderfully diverse place. CRISPR opens up many opportunities, both positive and negative. The benefits of curing disease, such as sickle cell anemia, and muscular atrophy, both of these caused by a single gene mutation. It can also increase the yield and durability of crops, slow aging, and reduce the effects of climate change by genetically engineering more CO2-hungry plants and trees. Another use can be reducing the effects of boredom, giving people more content lives. High levels of boredom also have the effect of reducing neuroplasticity, defined as the nervous system's ability to adapt itself in response to injury. Curing or even reducing boredom could therefore improve the mental well-being of many people for the better, and that is why boredom is sometimes described as the most dangerous emotion. Nearly all fields of life can be improved. even your most obscure, such as long-distance space travel. The sheer number of possible use is further exemplified by less vital, but still useful characteristic improvements, such as height, muscle mass, and lower susceptibility to disease, intelligence, as well as reduced levels of depression and boredom. Over the years, many studies have been done on the effects of continued boredom, the risk of Alzheimer's, petty crime, and all of these factors show how much potential CRISPR has to be a tool for good. CRISPR is introduced, normally with nucleofection, which is a pre-optimized delivery for DNA using electric pulses, to either a zygote or an embryo doing IVF, which affects all the host cells, and thus could in theory be passed on to its offspring. Due to the relatively small number of cells present, less CRISPR material is needed, and is more effective and cost-efficient than somatic, which only affects non-reproductive cells. However, like nearly all aspects of CRISPR Cas9, it is not without its downsides. In the book, A Cracking Creation, the author describes how the use of genetic editing of newborns may not be acceptable in many cultures and religions, and many might avoid science which they see as playing God. All of the techniques and processes aside, there is no doubt that CRISPR could fix many of life's greatest problems, death, disease and health to name few. But there's also the question of whether CRISPR is a Pandora's box. Once development is complete, will it lead to an inevitable evil? Throughout the video, I'll focus a lot of attention on the issues and problems of CRISPR, because the positives are so numerous will not fit in this same video. So it may seem like there's more dangers than there actually is. Firstly, there are obvious benefits of stopping and reducing the risk of rare diseases and ailments, even those as prevalent as cancer. Cancer occurs when a rogue cell loses the ability to destroy itself through incorrect transcription, and endlessly replicates itself, while draining vital nutrients from the blood and forming large tumours throughout the body. In the website that I recommend reading, City of Hope, the author describes the danger of cancer cells by describing how, most of the time, T cells, those responsible for destroying invading or otherwise not functioning cells, are unable to identify a tumour. However, CRISPR has a potential to modify them by removing the genes that may interfere with the T cell's ability to kill it. So far, the studies for this have only managed to temporarily slow the tumour's growth. However, there have been notable effects, and they still hold promise for the future. As well as improving crop yield and size, CRISPR can also prevent the negative effects of overpopulation by feeding the increasing population. An example of traits found in genes that's been used by humans to produce better fruits and vegetables are strawberries. Typically 1.3 centimeters in diameter, thousands of years of selective breeding has created crops of strawberries consistently 4 to 5 centimeters in diameter, a 4 times increase in size. Although larger crops do not necessarily mean they are more nutritious. Another use is aging. Every human experiences aging, some at different rates. This is accompanied by wrinkles, a greater susceptibility to disease, and partial muscular atrophy, the loss of muscles over time. However, some animals can in theory live forever, so what's stopping us from introducing this trait to humans? Some animals, such as a jellyfish, Croptosis dohrnii, lobsters, and many species of sea urchins simply do not age, or do so significantly slower than other creatures. The question is, why do they not age, and yet humans do? Aging itself is not the problem we should be looking at. It's the greater likelihood of damage which comes along with it which causes the issue. Over time, telomeres, the form of protective sheath which covers the end of chromosomes and cells, the chromosomes contain DNA after all, gradually get destroyed by environmental factors, such as sunlight and background radiation. With this gone, the cells start to make transcription errors, and gradually the DNA copied from one cell to another gets mutated. They become harmful to the body, or just stop working. In lobsters, their bodies produce a never-ending supply of telomerase, the enzyme responsible for creating telomeres. So is it as simple as introducing an enzyme? Well, no. The problem with lobsters is that this causes them to keep growing, as it requires massive amounts of energy. However, if this issue can be ironed out, then humans could theoretically live forever. An alternative solution would be to program human bone marrow to release a constant stream of stem cells throughout their life. Stem cells are cells which still have not specialised into more specific ones, such as red blood cells, heart cells, or liver cells. As such, this could stop the problem of old cells being degraded through time. The animal called a hydra has cells which do not go through senescence, which is the process of deterioration with age at all. This seems to be thanks to a set of genes called FOX0 genes. When researchers prevented the gene from functioning, the animal began to show signs of aging and would no longer regenerate as they once did. There's no doubt that this gene shows remarkable promise, but the problem of moving this gene into humans and retaining its functions still remains a problem. Another example of a positive trait is lowering metabolism rates enough to allow easy travel in space. The genes for hibernation are already found in humans, it's just a matter of switching them on. However, all of these scenarios already mentioned are only possible uses, are not limited to hypothetical scenarios. A recent example in the news of this is the world's first curing of T-cell acute lymphoblastic leukemia, otherwise leukemia. Normally incurable, the doctors decided to perform an experimental procedure in which donated T-cells were genetically edited to target cancer cells. This is the first time that a cancer treatment has successfully altered the building blocks of DNA. After 28 days and a bone marrow transplant, all signs of cancer were untraceable. Although the long-term effects are unknown, this shows how advantageous new technology could be to cure all forms of cancer. This treatment involves changing the genetic code of immune cells to hunt down and kill cancer cells while leaving themselves alive. Professor Wasim Kristeen, one of the scientists behind his treatment, has admitted that this was their most sophisticated cell engineering so far and paves the way for other treatments. Now, onto the dangers of CRISPR. One of the many ways CRISPR can be used negatively to impact humans would be scientists editing mosquitoes with a gene drive, which is a technology that, as described on the website Live Science, dramatically increases the likelihood that a particular suite of genes will be passed on to the next generation, allowing the genes to rapidly spread throughout the population and override natural selection. An example of this is introducing a gene for susceptibility to malaria into mosquito populations, which is made far easier, as this can already be found in small amounts. Another danger is that of inadvertent effects caused by lack of knowledge or incorrect protocols. An example of this is an incident caused by a company called Recombinetics. In October 2018, their unintended goal to genetically engineer dehorned dairy cows to reduce the danger posed with their long horns had unintended consequences. As part of the editing machinery, a piece of bacterial DNA that delivered the desired gene into the cells, called a plasmid, was accidentally copied into its genome. And this, in fact, was part bacteria. A teeny tiny part, although around 4,000 base pairs out of 3 billion. This bacterial slice included a few genes for antibiotic resistance commonly found in plasmids. This in itself was not problematic, but the fact that this transfer occurred in a lab and not in a natural scenario causes many red flags to be raised about the possibility of spreading antibacterial resistance. What made it worse even was the fact this error was not discovered by Recombinetics, but by the FDA, the American Food and Drug Administration, just before the product was released to the public. Moreover, Recombinetics petitioned the FDA to designate its gene editing cows as being generally recognised as safe, and as such not required to be tested by them. The whole technology is a new and unknown science. As such, there may be hidden consequences down the line. After all, rapid evolution on this scale is a phenomenon that had never occurred before, undoubtedly for a very good reason. There may be consequences which may not show up for maybe even multiple generations. Now, onto issues. There are many ethical issues that are associated with CRISPR. An example of this is religious concern. Many religions may not accept using CRISPR because it's playing God. This may cause anger towards the church if the only cure to a relative's illness is not accessible, not just because of income or location, but because of the church's religious beliefs. Perhaps causing a lack of trust in the church will be further exacerbated by the religious divide in society. People who have full access to CRISPR, able to genetically alter themselves to be not just more disease resistant and healthy, but physically and mentally superior. As well as that, the effects of removing what is perceived as undesirable traits will reduce biodiversity, which is a prime reason why multicellular life is even possible over singular, cellular, and asexual reproduction. Biodiversity means that even in the event of a catastrophe, a small segment of the population will survive and pass on their more successful genes to the offspring, ensuring an overall race will go on, whereas a biologically identical race will potentially all be wiped out by any large-scale events such as a dangerous pathogen or sudden, extreme environmental change. This already occurs in large monocultures of crops due to human involvement in selective breeding. If that isn't enough, then the lack of biodiversity can also cause problems of development and improvement. From the lack of different perspectives that your environment can bring for both humans and animals. One of the most prevalent concerns with CRISPR are designer babies. Designer babies is the process of genetically engineering non-essential traits purely for cosmetic appearance. This can range from something as simple as height, intelligence, and muscle mass to the more controversial topic of skin, eye, and hair colour. The question then posed is, what is the ideal child? The whole idea of prioritising one characteristic over another could easily lead to a social divide, racism, and discrimination against those who do not have what is seen as an ideal trait, and thus a decrease in biodiversity. The topic of cosmetic editing might lead to less development and essential uses for the technology, these being preventing diseases and negative conditions, as cosmetic traits are far harder, more expensive, and complicated to design or implement, as these traits are generally spread between genes and caused by hundreds of them, whereas disease susceptibility is generally caused by only a few genes in a set location. However, many people argue, rightly so, that designer babies are no different to cosmetic surgery as an adult. CRISPR is a Pandora's box. Once it has been opened, it is near impossible to stop. An example of this is a HIV twins controversy. On 26th November in 2018, Kai, a Chinese scientist, edited genomes of 2 embryo twins to give them HIV immunity. The parents took place an experimental procedure involving in vitro fertilization, and then the use of CRISPR Cas-9 gene editing. The embryo's genome were edited to remove the CCR5 gene in an attempt to confer resistance to HIV, HIV being human immunodeficiency virus. Normally, this would have been a scientific miracle. However, the experiment was done in secrecy, and the consent from the parents was incomplete and inadequate. The experiment was done in secret due to it being illegal for HIV-positive fathers to undergo IVF treatment in China. This clearly shows that access to technology without restrictions can allow people to do whatever they want. The possibility to eliminate HIV might have normally been seen as a miracle, and had the scientists followed the correct ethical and legal guidelines, it might well have been. CRISPR Cas9 is a technology that will become more commonplace, whether it is deemed dangerous or useful, as it allows an easier and more efficient way of editing genes than any technology before it. And any technology of this scale will always have drawbacks and problems with it, although these associated with CRISPR are often larger than any example that has come before it. I also believe that the reputation of this technology has been tarnished by some early adopters of it, such as the talked about Kai, who have clearly shown to the world that it can be used behind health organisations' back from malicious or unsafe regions. However, that's not to say that this event was a bad thing. In fact, I believe that this scenario, and independent scientists using gene editing to goals not allowed by laws and regulations, has actually helped raise awareness of the dangers and issues that may have not normally been discovered or raised, and thus this may have helped prevent a larger scale issue in the future. So then, that leads to the question of how do you ensure CRISPR is a tool for good? For CRISPR Cas9 to be so, I believe it must be shared equally, regardless of how wealthy the recipient is. For this purpose, it has to have at least 3 things. 1. Be created by non-profit organisations, such as the NHS or the World Health Organization, and have strict research regulations. CRISPR Cas9 would have to be created as a non-profit organisation, possibly backed by that individual's country's government or some organisation which branches off across all such countries, such as the World Health Organization. This would mean that individual developers seeking to make money off this technology would be guaranteed to be undercut by prices, and thus requiring them to get funding from an appropriately reliable organisation, thus stopping any dangerous or untested knockoff products. As well as that, CRISPR Cas9 has to be accessible, and also have strict research guidelines. This could be as simple as having regular checks on labs which use CRISPR, to make sure it is all ethically sound. Background checks on staff who wish to work there could be conducted to stop people such as Kai from illegally and perhaps unsafely editing and experimenting on embryos or other biological materials. Typically, only high-income countries would have the knowledge and technology to research CRISPR. This could create an even greater divide between rich and poor countries, as those with easy access to CRISPR will become not just economically stable, but physically superior, whereas lower-income countries will have to import the product, giving yet another reason for it to be a non-profit, to make it easier for countries with less money to get a product which will ultimately improve the standard of living of millions of people, such as giving HIV AIDS resistance in rural areas of places such as Africa. Overall, I believe the conclusion to whether CRISPR is a tool for good or a danger is simple. Without control and legislation limiting the use of CRISPR to key fields such as disease prevention and improving human well-being, CRISPR is guaranteed to be exploited and misused for personal finance. However, if full safety procedures and appropriate equal distribution of products are maintained, as well as some form of worldwide scientific consensus, taken from a wide audience to give an accurate representation of the public's opinion and scientific evidence, then I believe that CRISPR Cas9 is a tool for the betterment of humanity, to improve quality of life and standard of living for all. Like all technologies, there will always be those who will use it for personal gain, but it also poses opportunities to do good as well, to make changes in months that would have taken centuries to do normally. And that is a rare opportunity that cannot be passed up on, to improve the standard of living and quality of life for many people by curing diseases, increasing crop yield and efficiency for the foreseeable future. That is all. Thank you for watching. I'll see you later.